All right, so welcome back to the NIH Library Bibliometrics Training Series. My name is Chris Belter, and today we're going to be talking all about topic analysis. So given a set of a couple hundred to a couple thousand publications, what are those publications about? What are the major research topics that are represented in that publication set? How are those topics changing over time? All of those kinds of things. This is a pretty common question that we get in bibliometrics. Either we're looking at publications by a particular research group and we're interested in what that research group or institution tends to write papers on, or we're looking at a set of publications on a particular disease or a topic area like diabetes or Alzheimer's disease, and we're interested in looking at what the research looks like, how it's structured, what the major subtopics are within that larger topic area, those kinds of questions. So that's really what we're going to be focusing on today. And we're going to be focusing on quantitative methods of doing topic analysis. Obviously, there are all sorts of qualitative coding techniques that one could use to assign things to topics based on subject expertise. We're not really going to be talking about those kinds of things today. We're really just going to be focused on the automated methods of doing topic analysis. So we're really focused on sets of publications where that kind of qualitative coding just isn't practical. So we're looking at, 500, several thousand, tens of thousands of publications. That's really the scale that we're talking about. And so at those scales, doing that kind of qualitative coding just isn't practical anymore. So we're going to talk about how to do that in a semi-automated fashion. So I'll start by talking a little bit about some of the methods that we can actually use for doing topic analysis, because there are probably dozens, if not hundreds, of different approaches for doing this kind of work. So I'll give a quick introduction to what these kinds of approaches are, how they work, and just a couple of examples of some of those different methods, just so you've got an idea of sort of what's out there in the broader scheme of things. I will then zoom in to talk about two in particular, and those would be word co-occurrence networks and bibliographic coupling networks. Both of those are going to be building on all of the network concepts that we've been covering in the previous sessions, so it sort of follows on nicely from the previous couple of recordings to here, where we're really going to be using some of those network-based approaches for doing topic analysis. I'll be jumping back and forth between the PowerPoint presentation and live demos. So I'll give you a little bit of an introduction to the conceptual aspects of what this thing is, and then I'll jump into the hands-on portion to show you how to go from a Web of Science.isi file all the way through to visualizing the resulting network in Gephi. So a little bit, again, about some of these topic analysis methods. So within topic analysis, there are really two major families of approaches. There are text-based methods, and then there's citation-based methods. The text-based methods are the ones that you're probably the most familiar with. These are approaches that essentially use terms as indicators of relatedness. So the idea is that they use the same terms, or if papers use the same words, they are in some way related to one another. So this is sort of again, one of the more sort of intuitive ways of doing this kind of an approach. There are a huge number of text-based approaches for doing topic analysis. At the most basic level, we could just do term counts and say which terms appear most frequently in the document set to get an idea of what the papers are about. There are also word co-occurrence networks, which I will talk about in more detail a little bit later on. And then there's a whole family of text mining approaches, document clustering approaches, all kinds of things like that, where it's more in depth around analyzing the terms, sentiments, phrases, all kinds of things that actually appear within either the titles, the abstracts, or the full text of particular documents. Again, that's an entire family of approaches, which I'm not really going to talk about here today, but I just wanted to let you know that there are a huge number of those types of algorithms out there. That's kind of where they all fall in the grand scheme of things. The other major family of analysis approaches are actually citation-based methods. So with these types of approaches, we're actually using citations as indicators of relatedness between papers. So the idea is that if papers cite other papers, that's some indication that those papers are related to one another. The idea, again, being that you really only cite papers that are relevant to the paper that you're currently writing. So we can use these sort of citations as other means of connecting papers based on topical approaches. So there are, again, quite a family of approaches towards these citation-based approaches. I will talk about all of these when we actually get to the citation approach, but again, I just wanted to point out that there are at least 3 different methods of doing this kind of citation approach. There's direct citation approaches, there's bibliographic coupling, and then there's a method called co-citation. Again, I will explain what all of those are a little bit later on, give you some examples of what those look like, but again, those are just methods within this sort of larger umbrella of citation-based approaches for analyzing research topics within documents. So what I'm going to do today is I'll give you an example of one from each of those different families of approaches. I'll give you a word co-occurrence network from the text-based approaches, and then we'll talk about the bibliographic coupling approach from the citation-based method, so you've got an idea of how those two actually work. So the first thing that we'll talk about are these word co-occurrence networks. So within this kind of a network, the nodes represent terms that appear in documents, and those terms are connected to one another if they both occur in the same document. So if you've got a document that has a title that contains the phrase lung cancer, for instance, lung and cancer would be 2 of the nodes in the network, and they would be connected because they both appear in the same document. They both appear in that one title. So if you've got 15 documents that all use the terms lung and cancer, you could then build up a weighted network of those terms where lung and cancer would be connected to one another with a weight of 15, because they co-occur in 15 documents in the document set. So that's kind of what we're talking about here, is if terms both appear in the same document, they're connected, and then the weight of the connection, or the weight of the edge, is the number of times they co-occur in your particular document set, or the number of documents in which they co-occur in that larger document set. So there are both pros and cons to this approach. On the advantages of the word co-occurrence networks are they're very, very easy to make. Within either Sci2 or VosViewer, they are very, very easy to just sort of plug in and play. They are also very, very easy to understand. So when you're giving a visualization of a word co-occurrence network, it's very easy for your viewers to actually understand what you're looking at. Essentially, they end up looking very similar to a word cloud, which is something that people are already pretty familiar with looking at. But the advantage with word co-occurrence networks is that proximity actually indicates relatedness. So you can actually position terms related to one another closer together, so you can actually see clusters of terms within that document set, so that tends to correspond to the research topics in the document set. So it's very, very easy for your viewers to understand what it is that you're giving them and the visualization that you end up creating with this approach. They are also very accurate, at least broadly speaking. They tend to give you a very good idea of the general landscape of what these papers are about. They give you the very large research topics. They tend to be pretty accurate in terms of representing what those major clusters are. On the downside, though, they are somewhat high level. They're not necessarily as granular of topic areas as you might wish. So that kind of brings me into the disadvantages of this kind of an approach. So one of the most obvious disadvantages to this approach are synonyms and homonyms. So words that mean the same thing in different contexts, or if there are different ways of referring to the same concept, those things don't necessarily get taken into account in these particular settings. So if you're looking at a set of papers on cancer, you may have papers that refer to cancer as cancer, neoplasms, tumors, melanomas, all of these other different kinds of cancers that are all referred to the same concept, but are actually different terms or different ways of referring to that concept. So if you're just working with titles and abstracts, this is definitely a problem when you're going through and trying to analyze these topics. If you're doing word co-occurrence networks on controlled vocabulary terms, this is less of an issue, but that brings up its own set of problems as to how the controlled vocabulary is actually structured, but that's kind of a whole different ball of wax. As I mentioned before, the topics that tend to emerge tend to be relatively low in terms of the granularity. So they're very broad research topics. So they're topics like cancer or genetics or, I don't know, infectious diseases. They're very, very broad topic areas. And so in some cases, those are appropriate. In other cases, maybe they're not so appropriate. So it sort of depends on what your goals are for how detailed a topic structure you actually want to get as a result of your particular analysis. In some cases, it's fine. In some cases, it's not. And the final sort of disadvantage here is you don't get an assignment of a paper to a particular research topic. Again, we're just working with the terms here, so we can get a sense for what the terms are that appear in the document set, but you don't get to say that this document belongs to this topic area. You don't get that kind of level of detail. So if you're interested in looking at the number of papers in topic areas, for instance, or changes in the topic areas over time, those kinds of things, those are much, much harder to do with word co-occurrence networks than they are with other types of approaches. So again, with these word co-occurrence networks, it's great for getting a very high-level, very quick overview of what these top papers are about. But if you want more detailed kinds of looks at these documents, maybe this is not the approach to take. So creating them in the Science of Science tool looks broadly like this. You sort of load in your publication set. You do take a step called, which I'm referring to here as normalizing the words. I will talk about that more in detail, but essentially it's getting rid of terms that don't mean anything, cleaning up some of the terms, removing word endings, stuff like that, to make the actual word co-occurrence network a bit more accurate. You then create the network based on those normalized terms. You then have to prune the network pretty aggressively in order to get it to actually work. And then you go into the visualization step. So this pruning step is really necessary because these word co-occurrence networks end up being very dense. So if you can imagine, if you've got a set of 500 papers, those papers all have 20 words or maybe 10 words in the topics, you're already starting to talk about a very, very high number of terms and a very, very high number of co-occurrences. That's exacerbated if you end up using the abstracts. Again, if you've got 200 words per abstract, that's 200 terms with a co-occurrence of 1 with just one paper. Multiply that by 1,000 papers, that becomes very, very dense. So practically speaking, when we're creating word co-occurrence networks, we're really just creating them on the titles of papers. The abstracts are just too dense to get a whole lot of meaning out of. So when we're doing co-occurrence network or word co-occurrence networks, we really just focus on the titles because those titles have a much higher topic specificity than the abstract does. The abstract just has too many terms. For a word co-occurrence network, it just doesn't really work. If, however, you're doing text mining or topic modeling approaches, then we will both use the titles and abstracts because those other types of approaches are much better at handling those high volume, that high amount of data. And so some of those other approaches, you can take the abstract into account, but with word co-occurrence, you really can't. So again, just different methods of working with this data depending on what you have and what you're working with. So let's go ahead and jump into the live demo, and let me show you what this is actually going to look like. So for these live demos, I'm going to go back to the Science of Science tool, and I'm going to work with that .isi file that we've been working with throughout the session to actually end up working with this particular publication set. So the first thing I'm going to do is I'm going to load my data into the Science of Science tool. So I'm going to do File Load. I'm going to navigate to the particular file that I want to load. Select it, and then click on Open. Okay. Now that I've got it in here, I can again just work directly with this .isi file. So this is a little bit of a different workflow than we worked with the cleaned version where we were importing a CSV file. With this, we can work directly with the .isi file exactly as it was from Web of Science, because we're not dealing with authors or institutions. We're just dealing with the original data, so we can go back to this .isi file. So the first thing we're going to do when creating a word co-occurrence network is we're going to do that normalize word data step. So fortunately, the Science of Science tool actually builds this into the tool. So we can actually just run algorithms directly in the tool to take care of all of that work for us. So to do that, we're going to go up to pre-processing, and we're going to select topical, and then we're going to select this lowercase, tokenize, stem, and stop word text. So we're going to select that. And what this is going to do is it's going to transform all of the titles to lowercase, so capitalizations don't end up throwing off our matching algorithms. It's going to remove stop words, so words like of, and, the, by, those types of terms that don't actually have any meaning, but that appear very frequently in my document set. It's just going to go ahead and remove all of those. And it's going to do that using a particular stop word list that's already built into the Science of Science tool. So the Sci2 tool actually comes with its own stop word list, so you don't even have to worry about it. It's going to be automatically pre-filled in here, and so you can just leave this as is. If you want to customize that stop word list, you can do so, or if you wanted to bring up a custom stop word list, so if you wanted to add terms to that stop word list, you could do so in the Browse feature, bring in a custom stop word list, and work with it that way. The vast majority of the time, I don't do any of that. I just use the default stop word list and just go with it from there. It will also put a new separator between terms, so it'll put this pipe delimiter between all of the terms that actually it finds in the document set. So again, it sets it up to create this co-occurrence network, and then finally it stems the terms. So what stem does is it actually removes word endings. So endings like es, ing, ed, these other variants of terms, it will just go ahead and lop all of those word endings off the end of the terms so that cell and cells end up being matched together as the same term rather than being two different terms. So again, Sci2 is going to do all of this in this one step. It's pretty nice. So all that we really have to do here is select the particular column that we want to run this operation on. So in this case, we're going to scroll all the way down to near the end, and we're going to click the checkbox next to title. Again, we're going to be doing this word co-occurrence network on just the article titles, so we're just going to run this on the titles and ignore everything else. So we're going to click on title and then click on OK. And then very, very quickly, it's going to run, and it's going to give you this with normalized title data set. So this is what we can now create the word co-occurrence network on. Again, it didn't touch my original data, but it gave me this new data set that I can work with and then create a co-occurrence network on. So with that selected in the data manager, we can go now to data preparation and then click on extract word co-occurrence network. So again, this is one of those networks that's already built into the tool. It's already set up to do nearly all of the work that we need, so we can just go ahead and run this particular algorithm as we go. So I'm going to select that. And again, we're going to get another pop-up box. This is just, again, the way that Sci2 works. So what's interesting here is we actually have to select a node identification column. This is the only algorithm where we have to do this, and I don't really understand why, but we do. We have to select some column that uniquely identifies all of the publications in our data set. So we can just select either the Cite Me As field, which Sci2 created. We could select the UT, which is the unique identification number, basically any column that appears in all of the rows in the data set and is unique to each of the rows. So I'm just going to select Cite Me As. And you do need to select something here, because if you don't select anything, it's not going to actually run properly. You can also select the text delimiter. Again, we're just going to use this pipe because that's what we used. That's what the previous step actually put as the delimiter, so we're just going to leave that alone. I will also mention that there's an option for using an aggregate function file here. Again, we're not going to use it, but if we wanted to calculate additional attributes about the terms that appear in our network, we could again use that here. We're not going to use it, so we'll just use this as is. And then again, the only thing we have to do here is scroll down to the bottom and select the particular column that we actually want to run this on, which is our title column. So again, we select, or we go all the way down to the bottom, click that checkbox next to title, and then click on OK. So it takes a minute to run, and then it creates this word co-occurrence network. To give you an idea of just how dense this is just from the title, we can go ahead and run analysis networks and network analysis toolkit just to get a sense for how big this network actually is. And it's going to take a while because there's a lot of edges in this uncut network. So in this particular set of documents, we've got 2,000 nodes and we've got 29,000 edges. So just using the titles of these papers, we've got a very, very dense network already. So we, again, want to prune this network in order to make sense of it, to pull out terms that only co-occur in a very large number of documents in our particular document set. So again, after running the network analysis toolkit, we want to go back to the data manager, select the co-occurrence network, and then we're going to go up and start removing some of these edges. So we'll do pre-processing, and then networks, and then dichotomize, just like we did with the co-authorship networks to do this pruning. So we'll select dichotomize, and then we'll set a threshold for how much we actually want to cut. Typically, I will start with somewhere around 5 and see what it looks like, and then maybe play around with different thresholds from there to see what it actually ends up with. So I might do weight greater than 4 just to get a sense for how big the network ends up being, and then I might set it higher or lower. Another thing that I find helpful is sometimes if I want to be really selective in this, I will set it to greater than around 1% of the documents in my document set. So in this case, if I've got 900 documents, I might have it set it to weight greater than 8 in order to only keep terms that co-occur in at least 1% of the documents in my data set. That tends to be relatively restrictive with smaller data sets, so that's why I typically start with weight greater than 4, but you might play around with different thresholds, again, just to see what that looks like, how many nodes you end up working with, that kind of thing. I tend to try and shoot for somewhere around 100 to 200 terms in the final word co-occurrence network. I tend to find that more than about 200 terms seems to be more than can be adequately visualized in a particular setting, so that's why I tend to shoot for 100 to 200 terms. But again, you might play around with different thresholds. But for this particular purpose, I'll set weight greater than 4 and then click on OK. So now that I've removed those edges, I again need to remove any of those islands and additional sets of terms that aren't connected to anything else. So I'll go up and do analysis, networks, unweighted and undirected, weak component clustering, to actually be able to just pull out the largest connected component. Because again, with these word co-occurrence networks, all we're really interested in is the largest set of terms that are all connected to one another. So that's what we'll do here. So again, just like we've done in some of the previous settings, we'll just run this weak component clustering to pull out just the top one cluster. So we'll set that to 1 and then select OK. So this gives me a weak component cluster of 185 nodes. So that's pretty good. That's about what we're kind of shooting for. If we wanted to reduce this further, we could set it higher or lower depending on what we wanted, but this looks pretty good, so let's go ahead and go with this one and actually move into the visualization steps. So just like with the co-authorship networks, we can go up to visualization, networks, and then select Gephi. Just like with the import reports, with the co-authorship networks, we get this import report. I've got 185 nodes, 500 edges. That's nice. No issues with the import. So we'll just click on OK, and we'll work with it from here. Center this. So just like before, this is going to look very, very similar to the visualization steps we did with the co-authorship networks. We're going to apply a layout. Then we're going to do some sizing and coloring of the nodes. We're going to do some additional steps in here. We're actually going to look for clusters of terms that are more connected to each other than to other clusters to try and get a sense for where those major research topics are. And then we'll go over to the preview tab to set our final visualization settings. So we'll start out with the layout tab. So we'll go from choose layout. I'm going to select the Force Atlas algorithm here. And I'm also going to go ahead and reduce the attraction strength. If I ran this as is, it would basically end up being just a very small dot, and we wouldn't be able to actually see anything. So I'm going to go ahead and reset this from the outset to get a more meaningful layout. So instead of having this attraction strength as 10, I'm going to reduce it to 0.1. And again, if it doesn't allow you to actually edit this, go over to the data laboratory, come back to the overview tab, and then try again, and it should allow you to edit. So with this setting, I'm going to go ahead and run the algorithm. And then I'm going to wait for it to stop moving around to stop it. All right, there we go. And then I'm going to click stop to stop the algorithm from running. So again, I can use the mouse wheel to zoom in, right click and drag to center, and so forth, so I can sort of get a sense for where my network actually is in the larger visualization. So now that I've got the base layout done, I can now go ahead and start doing some sizing and coloring of the nodes and edges to bring out some of the attributes that are already present in my network. So I'm going to go up here to the Appearance tab. I'm going to shift from color. I'm going to go straight to size. So I'm going to select the Size tab. And I'm going to size. I'm going to select Ranking. And again, make sure you've got nodes selected and not edges. So nodes, size by ranking. And then we're going to select references as the attributes we want to size by. I'm going to use that same 5 to 50 scale. You could play with it just to see, but I'm going to use this, and then click on Apply. So now we've got some of the sizes coming out, and we can see some of these terms are much more frequently occurring than others, but we can also start to see groups of terms that start to appear within this larger word co-occurrence network. The next thing that I'm going to do is I'm going to color code the edges based on the number of co-occurrences, or how frequently these terms co-occur within my document set. So I'm going to go back to Appearance, I'm going to select Edges, I'm going to select Ranking, and then in the drop-down, I'm going to select Weight. So again, it's going to bring up this color scale. I'm going to click on the little box next to the color scale to bring up a recent color scheme that I'm going to use. And then in this case, I'm going to use this grayscale that I've worked with because I want to color code the nodes based on the clusters that appear. So I don't want to use colors in the edges because that would then distract from the colors in my node set. So that's why I'm going to use this grayscale to represent the frequency so that it doesn't necessarily get in the way of the nodes. So select this grayscale and click on apply to get a sense for where some of those major co-occurrence terms are. The next thing that we're going to do, and the thing that's a little bit different from what we did in the co-authorship network, is we're going to run an algorithm to identify clusters of nodes. So remember back when I first was introducing this network analysis concept, is that networks tend to be comprised of subnetworks, or so there are groups of nodes within any given network that are more densely connected to each other than they are to other nodes in the network. So there are actually algorithms that have been designed for finding these clusters. So we can run just a set algorithm on any particular network we happen to have, and it will go through and find these clusters of nodes, identify them, and then actually assign them to particular numbers so that we can actually use those attributes later on to do some of this color scale work. So what we can do is we can go over here to statistics over here on the right, and you can see that there's a modularity statistic. So this is what we're actually going to run to find those clusters of nodes. So to do that, we're just going to click on this run box next to modularity. And that's again going to bring up a little pop-up box where we can tweak the algorithm if we want to, though nearly 95% of the time I leave this as is. So just click on run there and then just click on OK. So it's going to run, and then it's going to give us this report of what the algorithm actually found. So in this case, it says it found 10 communities within this set of documents. So these are 10 clusters of terms that appear more frequently with each other than they do with other nodes in the network. If you're following along, you may actually get different numbers of communities with the same document set. And sometimes you will actually get different numbers of communities if you rerun the thing. So this is actually based on the actual algorithm itself. The algorithm tries to determine how many communities to find given the particular document set that you have or the particular network that you're working with. But it has some random features built into it. So again, because there are some of these random features, it will sometimes give you slightly different numbers of topics. So in some cases, you might find 8 communities, it might find 9, it might find 11. Most of the time, though, it's going to find either 10 or 9 or somewhere within that set. And if it finds any additional communities, there's usually maybe 2, 3 nodes within there. So it's not going to substantially affect your overall results. It's just going to tweak maybe one or a couple of nodes. Also within this modularity report, you scroll down to the bottom, there's a citation to the actual algorithm that it's running. So if you want to document which algorithm you're actually using to find these communities, it has a citation to that. It also has a citation to the particular implementation of that algorithm within Gephi. So again, you can sort of say, this is a particular algorithm that I'm running to find these communities. So most of the time, though, I just glance at the number of communities and then click on close. So the network doesn't look very different now, but if we go up to the data laboratory, we can actually see that it's actually added a new column to the nodes, which is this modularity class. So that modularity class is actually the results of the algorithm we just ran. So it assigned every single node to a particular modularity class and then added that number to the node attributes so that we can actually work with this in the sizing and coloring operations. So we can go back over to overview, and now if we want to color code based on those modularity classes, we can do so. So we can go back up to appearance, we can go to nodes, and then we can select the color option now because we want to color code by those modularity classes that the algorithm actually found. So we'll do that. And then instead of clicking on ranking, we're actually going to click on partition now. So what we want to do is we want to apply this sort of qualitative or categorical scheme to the nodes in the network. And so we're going to use the partition option to do that. So again, we'll select partition, and then the attribute we're going to use is this modularity class. So that will bring up a color scheme and a set of colors that if we wanted to just go ahead and apply this, we could use this as is, or again, we could click and hold on any one of these, drag it around to actually change the color scheme or the color of any particular category and go with it as is. We want to just see what this looks like, we can just click on apply. So we can see that now that we can see some of these clusters of terms that tend to appear together. So again, we've got clusters here, got sort of a cluster in the middle, some others up here, so on and so forth. So we can start to get the sense for these groups of terms that tend to appear very frequently together in documents in the document set. If we want to change all of these colors all at once, say we don't like this pastel color scheme, there's another thing that's kind of buried in the interface, which is this palette link. So if we click on that, it will bring up color palettes that maybe you've used before, or it allows you to select color palettes from here, or it allows you to generate new color palettes. And so this is what I typically do, is I'll go over here to generate. And then there are a number of preset options for generating color palettes. So let's, if I click on the down arrow here, I'll select maybe the intense color palette, and then it will also allow me to set the number of values. So remember, we've got 10 clusters in my data set. I want to actually end up with 10 different colors in this color palette. So I want to either uncheck the box next to limit number of colors, or increase the number of colors to 10 so that we have enough colors for that particular document set that we're working with. Then when we have that, we can click on Generate. And so that will generate a color palette that we can use to apply as is, or we could keep clicking on Generate to generate different color palettes. So we kind of just keep clicking on it until we get a color palette that we like, and then click on OK, and it will use that particular color palette in the actual visualization. So let's go ahead and click on OK, and again, it will use this particular color palette that I just generated, and then click on Apply to actually apply it to the actual visualization in my actual network. So the next step that we're going to do is, once we've got these colors, is to actually look at the node labels that, again, just like the co-authorship network. So we're going to click on the little T down here at the bottom. And then we're going to do the size mode as node size. I'm going to adjust this just a little bit so we can see. And now we can start getting a sense for what the topics actually are. So we zoom in here, we can see there's quite a lot around pandemic influenza, influenza mortality, maybe seasonal aspects of influenza, but essentially a group of papers around influenza. We see that there's a group around virus transmission, maybe transmission dynamics, maybe transmission in populations, those types of things. Disease and model, so maybe disease modeling. So modeling, maybe modeling the transmission of diseases in particular networks. We can see there's also a cluster over here around malaria. There's a cluster up here around vaccines, maybe rotavirus vaccines, so on and so forth. So we can start to get a sense for what the major research topics are in this particular publication set just by some of these clusters of terms that tend to co-occur in documents in my document set. So again, we can sort of use this as a way to get a very quick sense for what the major research topics are. So from here, we would then go over, maybe run a couple more things to either rotate, label adjust, do some other tweaking that we did in the co-authorship networks, and then go over to the preview tab to set the final visualizations, settings, and then export an SVG file format that we can open in Inkscape like we did with the co-authorship networks. So from here, the process is nearly identical to what we went over with the co-authorship networks, so I'm not going to go over it here today, but again, that would be the general process. We would again tweak it, export the final visualizations from Gephi as an SVG, clean it up in Inkscape, and then export the final visualization from Inkscape into a graphic that we would actually work with in a report or a PowerPoint template or something like that. For the purposes of this course, though, we're going to go ahead and shift gears now, and we're going to start talking about the other major approach, which is bibliographic coupling. So let me close out of Gephi, come back to my PowerPoint presentation, and pick it up here. So that was all about word co-occurrence networks, and so that's kind of an idea of the text-based text mining approaches. We're now going to shift gears over into the citation-based methods. And so we're going to talk a little bit about what these citation-based methods are, talk a little bit about bibliographic coupling networks in particular, and then go through the same kind of a process to show you what it's actually going to look like for creating one of these bibliographic coupling networks using the Science tool in Gephi. So as I mentioned, with these citation-based methods, these are methods of using citation networks as indications of similarities between documents, with the idea that papers that cite each other tend to be related to one another. So to just kind of give you a sense for what this looks like, there are 3 different approaches within citation-based methods. The first of these is this direct citation approach. So what we can do is just use the direct citations to create a network that we can then cluster. So in this case, we've got a set of 4 documents, very creatively named A, B, C, and D. And we've got a series of citation relationships among them. So paper A cites paper C, and paper B cites both C and D. So within this direct citation network, we can actually create this and then use that same modularity algorithm to cluster this network as it is. And so if there are clusters of documents that all cite each other, that's an indication that those papers are all related to each other in some way. So that would be the most sort of common or simple approach to using citations for topic analysis, is create one of these direct citation networks and then cluster it to look for clusters of papers that all cite each other. However, in the literature, there are additional ways of creating additional networks based on this network structure. So based on the direct citation network, there are actually additional networks that we can create that sort of modify these citation behaviors. So we can use this citation network to actually create additional links between papers, even if they don't actually cite one another. So one of the elaborations of this direct citation network is a network called bibliographic coupling. So the idea with bibliographic coupling is that we're going to connect documents if they share references with one another. So if 2 documents cite the same previous document, that's an indication that the 2 documents are in some way related to one another. So in the diagram here, since both paper A and paper B both cite paper C, that's an indication that A and B are some way related to one another, even though there's no direct citation link between A and B. But because they both refer to C, there's some indication that A and B are related to one another. So that's what bibliographic coupling is it connects papers based on shared references. One of the advantages of this approach is that the more papers that two papers have in common, the more likely they are to be related to one another. So if A and B have 5 references in common, that means they're probably more related to each other than they are to papers that they maybe only share 1 reference with. So we can actually have degrees of relatedness within this sort of larger structure, whereas with a direct citation network, you can't really do that. It's there or it's not. Whereas with bibliographic coupling, you can actually weight edges based on the number of shared references. The third approach to working with citation networks is conceptually the opposite of bibliographic coupling, which is called co-citation. So with co-citation, papers are connected if they are cited alongside one another in a subsequent document. So again, within the diagram, because papers C and D are both cited by paper B, that's an indication that C and D are in some way related to one another. So again, because C and D are co-cited by B, that would then create a link between C and D. So conceptually, it's the opposite of bibliographic coupling, where papers are connected if they are cited alongside one another in subsequent documents. So bibliographic coupling and co-citation are just sort of different sides of the same coin, where we're using citations to create links between documents where maybe there aren't citation links that currently exist. But they do so in very, very different ways, and they give you slightly different looks at the same set of documents. So with bibliographic coupling, what's interesting is the links that are created with bibliographic coupling are fixed in time. They don't change. Because they're based on references, they're sort of retrospective, and so something is then connected to a previous document, and that connection stays even if you add new documents to the network. Whereas with co-citation, the links are actually dynamic. They actually change over time. So C and D may be linked at one point, but then maybe if we look 10 years in the future, maybe we see a new link between C and something else. So links can be added to C and D, whereas with bibliographic coupling, those links can't necessarily be added. From a practical standpoint, though, there's also very large distinctions between the two in terms of how many papers you can actually get in a resulting network. With co-citation, links are dependent on whether or not papers have actually been cited. So in order for a paper to actually be included in a co-citation network, it has to have been cited at least once. So if we're looking at very recent sets of documents, that's not necessarily the case. Or if we're looking at sets of documents that move relatively slowly, not all of the documents in your document set are going to have citations or are going to have been cited, so they're going to drop out of a co-citation network. With a bibliographic coupling network, on the other hand, you don't necessarily have that problem because you're relying on references rather than papers being cited. So because papers have references immediately when they're published, you can go ahead and create a bibliographic coupling network directly on those very, very recent publications without losing papers. So it doesn't rely on them having actually been cited. You can go ahead and you can actually include them in the bibliographic coupling network. So from a practical standpoint, we tend to focus on bibliographic coupling just because we tend to get more of the documents in our document set in the resulting network because we're not relying on them to having accumulated citations. So that's why we're focusing on bibliographic coupling here, because we can get more of the documents in our document set into the network. We can't get all of them into the network because some of them don't share references with any of the other documents in the document set. That happens. Sometimes we'll also lose documents if they if we prune this bibliographic coupling network, so if we're only interested in papers that share 2 or more references with one another, if papers only share 1 reference, they will then get dropped from the bibliographic coupling network. So as we're working with these networks, we do tend to lose some amount of papers over time. That's just a result of this particular approach. So again, with a basic bibliographic coupling network, the individual nodes are these papers in our document set, and they're connected based on shared references. And again, this is a weighted network because they're weighted based on the number of references 2 papers have in common. So again, we can sort of represent this as a weighted network and then use those weights to prune to get to the more meaningful sets of links between documents. Again, some pros and cons about these bibliographic coupling networks. On the advantages, we get much higher granularity out of these bibliographic coupling networks. So the topic areas that emerge tend to be much more specific. They tend to be more granular in terms of the actual topics that emerge. So we see much more detailed topics emerge from these particular approaches. We do also get papers assigned to documents with this approach. So when we run this modularity class with a bibliographic coupling network, we get papers assigned to particular topic areas. So we can then go back in and say, this is the number of papers on this topic versus this topic to get a sense for the emphases in the larger publication set. We can also do additional things to say who are the authors in each one of these publication areas, how are these topic areas changing over time, all of those types of additional analyses that we can build onto that initial topic set of assignments. So there's much more we can do with this, the result of this approach, than there were with the word co-occurrence networks. We also allow the papers to sort of tell us what they're about, which is kind of a nice approach to this. So rather than sort of using the terminology or somehow using some kind of top-down organization scheme, we're literally allowing the papers to tell us what they're about by the references they actually cite. So we're allowing the papers to say, this is about this topic because I'm referring to all of these other papers that are also about this particular topic. So it's much more of a sort of bottom-up approach to doing topic analysis than some of the other approaches are, which is kind of nice. However, there are definitely some limitations to this approach. One is that they are somewhat more difficult to create. There's actually a 2-step process for actually creating these networks. So it's a little bit more involved in actually creating these things. It also relies on very, very accurate metadata to actually do this. So in order to create links from one paper to another paper, those citations have to be in exactly a standard format. So if you're getting data from a different data source and you don't have consistent citation data, you basically can't do this. So if you're trying to do this from, say, grant abstracts, for instance, or grant records that cite publications, those citations are not in a consistent format across the entire data set. So practically speaking, you just can't do this unless you do some kind of additional steps to normalize those citations into a very, very consistent citation format. So it's limited in the data sources that you can actually run this on. They are also more difficult to sort of understand for your audience. So when you're presenting the results of this type of an approach, it requires more elaboration and explanation to actually get somebody to understand what the result of this thing actually is and how it looks. There's usually quite a lot of supplementary information that has to go alongside one of these visualizations because people aren't used to looking at bibliographic coupling networks. People don't even necessarily know what they are, so you have to explain the whole process and then explain what the visualization looks like in order to actually convey the information. So there's more work involved for your viewers to actually understand what they're looking at. And as I mentioned, you do lose papers along the way. So again, since you're relying on shared references, you're going to lose some percentage of the papers because they just don't share references at all, or they don't share enough references with other papers in the document set to be included in the final visualization. Typically, we tend to shoot for above 80% of the documents in the document set, but you are going to lose 10 to 15 to sometimes 20% of the documents, so it's worth keeping that in mind when you're presenting these results to people, to say that you are losing some data along the way in order to get to these research topics. Okay, so creating them in base Sci2, or sort of the standard format for the standard workflow for creating these is to load the data into the Web of Science tool. You create a paper citation network first, and then you create a bibliographic coupling network based on that citation network. So it's actually a two-step process. You create the direct citation network first, and then you run the bibliographic coupling network on that direct citation network to actually create the bibliographic coupling network. So that's what I mean by a two-step process, is you create one network first, you then create a network on top of that network, and then you can actually work with it. Again, then, if necessary, you can then remove some of those edges to focus on the most meaningful links between documents, and then go over into the visualization step. The limitation to this approach is when you're creating a paper citation network in Sci2, you don't necessarily get all of the information that you want in the resulting network. So when you do this in the base Sci2 operations, you don't, for example, get the titles of the papers in your final visualization. You only get a very select amount of information. The problem with that is when you create the clusters in that clustering algorithm, the algorithm doesn't tell you what the topics are. It only tells you what the numbers are. So it says, the algorithm says, I found a group of papers. They're all connected to one another, and I'm going to call them topic 2. It doesn't give you any more information than that. So it says there's this topic, it's topic 2. These are the papers that are assigned to topic 2, but it doesn't tell you what topic 2 actually is. You have to actually go in and figure out why the algorithm actually clustered that set of documents together. And you have to actually name the topic to something that's actually meaningful. So there is definitely a qualitative aspect to this whole process where you use the algorithm to determine what the clusters are, but you then have to go in and actually name the clusters to understand why the algorithm actually clustered them the way that it did. So there is this additional step necessary to actually figure out what the topics are, to actually name those topics. So to do so, you need some more information about what those papers are beyond just the citation. You need the titles. In some cases, you need the abstracts. You need some of that additional information. And if you're creating a bibliographic coupling network in the base Sci2, you don't have that information. You can go back and get it after it's over. So you can create it in base Sci2. You can visualize it in Gephi and export the node table from Gephi, and then you do a VLOOKUP operation to bring in the titles and abstracts of the papers that were in your original data set, but it's kind of a pain. So the way that we tend to do it is we actually bring those in using a custom aggregate function file, so we have those titles to begin with. So to do that, what we do is, again, we use these aggregate function files to bring those titles in. So I mentioned aggregate function files before, but I just wanted to quickly go back to them just to sort of explain what's actually going on here behind the scenes. So these aggregate function files are, again, custom functions to add attributes to a network in Sci2. So they're ways to customize what the network attributes actually are in a network that you create in the Science of Science tool. So they have a very particular file format, which I talked a little bit about in a previous session. They are either node, dot attribute name, then equals the column name that you're going to get it from, and then the function that you're going to run. So examples of this, if we want to bring in the titles of the articles in my data set, I would have a node attribute. So I would do node dot whatever we're going to call this attribute in the final network. In this case, we're going to call it article title. And then that's going to be equal to the title column from my original data set. And we're going to do dot mode here to calculate the mode of the article title. It's kind of a hack a little bit, but essentially what it does is it brings in the most common occurrence for each node in the network. So since papers are going to be the nodes and papers only have one title, calculating the mode of the article title just brings in the article title directly as it is. So that's what we're going to be doing. And it got other things we can do. Time cited is time cited.sum, other types of operations like that to bring in some of these additional attributes into the network. So working with them in this context then, we would then load the data into Sci2. We would then create a custom directed citation network using that aggregate function file. So again, we're going to basically create a paper citation network, but we're going to use that aggregate function file rather than the built-in sessions or the built-in function files that are built into the Sci2 tool. So we're going to mimic that paper citation network, just bringing in additional node attributes to be able to work with them in the final visualization. We're then going to create that bibliographic coupling network. We're going to prune it if we need to, and then we're going to go into the visualization. So again, it's a similar process to doing it in base Sci2. It's just a slightly different approach to bring in some of those additional attributes that we need to work with in analyzing the final results. So that's the workflow that I'll go ahead and demonstrate here with the publication set that we've worked with. So I'm going to return to Sci2 to do this. And I'm also going to return to that number of unique ISI records. So again, in the data manager, go back and make sure that unique ISI records is selected. And then I'm going to start a new process from here. So the first thing we're going to do is we're going to create that paper citation network. So we can go up to data preparation. And if we wanted to use just the base Sci2 operations, there is an extracted paper citation network that's built into the Science tool. We could run that and then run the bibliographic coupling network, and it would work just fine. So if you wanted to use just the built-in operations, that would be the way that you do things. However, what we're going to do is we're going to extract a directed network instead, because we want to actually use the aggregate function file that we have for this particular purpose. So I'm going to click on Extract Directed Network. And so we're going to select the source column and the target column. Remember, this is a directed network, so there is a source and a target, or a direction in this case. And so what we're going to do is we're actually going to do it backwards from the act of citing. So it's backwards from what you would probably expect. So what we're going to do is we're going to use the source column is actually going to be the cited references. And then the target column is going to be the cite me as field. So we're going to create a directed network from the citation to the citing article. So we're tracing the information flow rather than the act of citation. That's just the way that Sci2 does things. There are other tools that do this the other way, so it's important to sort of figure out which way your actual tool does things. The Sci2 tool does things from the cited reference to the citing article, backwards from what you're expecting, so make sure that you've got these two columns in the right order. And again, that cite me as column is a column that Sci2 created on import. So It's a unique citation that's in the same citation format as the cited references. So we can use that to build up this network of cited references. We can then select a different text delimiter if we want to, but again, because we're importing from Web of Science, we're just going to leave this as a pipe delimiter. And then we are going to go ahead and use an aggregate function file here. So to do that, we're going to click on Browse, and we're going to browse to the particular aggregate function file that we're going to use for this particular operation. So I'm going to go in to my folder full of aggregate function files. And in this case, I'm going to use this paper citation network plus title. So this is going to mimic everything that I get from the paper citation network, but it's also going to bring in some additional node attributes, including both the title and the year of publication. So I've got all of the information that I would commonly work with already built into the network from the beginning, so that when I export the node table later on, I have all of the attributes that I need. So I'm going to select that, then click on Open, and then I'm going to click on OK. So now it comes up with this network from directed edges, tells you a little bit about the network it just created. So now from here, now I can go ahead and create that bibliographic coupling network. So with that network selected, I'm going to go back to data preparation, and then I'm going to select this extract reference co-occurrence or bibliographic coupling network. So again, these networks are in fact built into the Science of Science tool. If we wanted to, we could also create a co-citation network from this document set, so that's also built in. So if we wanted to do a co-citation network, it's there. But again, for this particular analysis, we're going to go for the bibliographic coupling network. So we're just going to go ahead and select that. This is one of the rare algorithms that doesn't actually have a pop-up box associated with it. So it's just going to go ahead and run, and then it's going to go ahead and give me this new bibliographic coupling similarity network. One of the things to know about this network is because it was created from that paper citation network, it still has all of the nodes from that original paper citation network. So it has both the documents from my original document set and all of the references that they cited. But because those cited references don't share any references with anything else, they are now isolates in this bibliographic coupling network. So the first thing that you do after creating a bibliographic coupling network is always to just go ahead and delete isolates. There are usually tens of thousands of them in any of these bibliographic coupling networks. So if you try and run anything else on it without deleting the isolates, it's going to go really, really slowly because it's going to try and go through all those tens of thousands of nodes. So again, whenever you create a bibliographic coupling network, the first thing that you always do is go up to preprocessing, networks, and then run delete isolates to just go ahead and get rid of all of those to just speed up everything in your operations. So in this case, we started with 920 records, and we just removed 26,000 isolate nodes. So again, this is pretty similar when you're creating these bibliographic coupling networks. You always want to get rid of those to speed up anything else that you do to this network. So now that I've got this created, I obviously want to have a little bit of a look at this network to see how many papers I currently have, a little bit about the edge attributes, things like that, to see whether or not I actually need to prune this in any way to focus it in on stronger connections. This is, in some cases, this is necessary, in some cases it isn't. So this is one of those times you want to play around a little bit to find out whether or not you need to cut, and then if so, how much. So to do that, we're going to go to Analysis, Networks, and run that Network Analysis Toolkit to get some basic information about this particular network we just created. So again, it's going to run for a little while and then come up with some information about this network. So we've got 864 nodes in this network. So we're already down from that original 920, we're already down to 864. So we've already lost a fair number of articles just with this bibliographic coupling approach. So if we want to do any more pruning, we're going to lose even more articles. So with the pruning steps, it's always a trade-off between accuracy and representativeness. So the more you prune, or the higher the threshold you set for the edge weights, the more accurate the clusters are going to end up being. But the downside is the higher the threshold is, the more papers you end up losing. So if you set that above 1, you're going to lose more papers. And if you set that above 2, you're going to lose even more papers. So there's always this sort of trade-off between accuracy and losing publications. So what we tend to shoot for is, again, we try and get at least 80% of the publications from the original document set in the final bibliographic coupling network. So in some cases, we don't want to cut it all. In some cases, we do, though. We also can see that there are some node attributes that we have. So as I mentioned, I've got this year published attribute. We also have the article title, we have the citation count, and the number of references cited. So we've got some additional information about these publications from that aggregate function file. We also have 31,000 edges connecting these 864 documents. So if we try and visualize this as is, it may be far too dense for anybody to make any sense of. So we may need to go ahead and prune things. So we need to prune a little bit, but we want to be selective in how much we prune in order to not lose any more documents that we already have, or not as many documents, so that we lose as few documents as we possibly can. So one of the things we can do then is we can just calculate just the proportion of documents we already have that are present in the network. So we've got 864 divided by 920 so we currently have about 94% of the publications in from the original document set. So that's pretty good, but we could theoretically cut and see what it looks like if we set that threshold a little bit higher. So to do that, let's go back to Sci2, go to the data manager, select the with isolates removed, and then run preprocessing networks and then dichotomize to do that edge pruning. So again, it's going to churn through all 31,000 edges to get the edge weights, so it takes a little while, then get a pop-up box. So I want to keep edges with weight greater than 1. So I'm going to be very conservative in how much I remove here, again, because I want to be careful about removing too many documents. So let's click on OK. And then I've got edges with weight greater than 1. So now what I want to do then is remove any of those additional isolates that I just created, and then focus it in on just the largest connected component. This time we can just do this all in one fell swoop by doing analysis, networks, unweighted and undirected, weak component clustering, just to pull out the largest weak component of the network, pull out the top one, and then click on OK. So now I've got a weak component cluster of 788 nodes. So again, we're losing papers at a pretty rapid pace here. So again, just to get a sense for what we're working with, we can do 788 divided by 920 is about 86% of the nodes in the network. So for some analyses, 86% is probably okay. That's probably good enough for analysis purposes. So you could go ahead and go with this, or you could go back to the original bibliographic coupling network if you wanted a higher proportion of the papers included in the network and visualize that. It's up to you, and it also is up to the goals of the analysis. If you do work with the pruned version, you'll want to note that you did remove edges, and you removed papers, and that the final topic analysis includes only about 86% of the papers in the analysis, so that people are aware that that is, in fact, a limitation of this particular approach. So for the purposes of this course, I'm going to go ahead and go with this weak component of cluster of 788 publications. But again, if you wanted to, you could go back to the bibliographic coupling network and work with that. It's up to you. So with this, we're going to go ahead and go up to visualization, networks, and then Gephi. And from here, it's going to be very, very similar to what we just did in the word co-occurrence networks. So we'll start by glancing at the import report. We've got 788 nodes, and we've got 12,000 edges here in our particular network. So let's click on OK. And we get a really big blob. So again, first thing that we're going to do is apply a layout. So since we're working with a very high number of edges here, I'm going to use the Force Atlas 2 algorithm because it's better able to handle these very dense networks. So that's the one that I'm going to use. So I'll do layout will be Force Atlas 2, and then I'll just go ahead and run it as is to see what we get. So we'll wait for it to finish moving around and then click stop. So we can see that there's already some topic structure emerging here. So we see there's a bunch of papers over here. There's kind of an island of papers sort of heading out this direction. There's another cluster here, another one down here, so on and so forth. Again, we can sort of work with this a little bit to tweak the layout if we wanted to, but I'm going to go ahead and jump right into the sizing and coloring operations. So let's go into the nodes, and so we can size the nodes based on the number of times cited. So just to get a sense for where the most highly cited papers are in this particular document set. So we can do nodes, then size, ranking, and then the attribute, we'll use this global citation count, which is just the number of times cited, and then click on apply to apply the scheme. So it's hard to see currently, but it's now resized the nodes so we can get a sense for where the most highly cited papers are. We can also then color code the edges. Again, I'm going to want to use a grayscale to represent longer or stronger or weaker ties because I want to use the node colors to represent those clusters that I'm going to find. So I'm going to use a grayscale here. So I'll do edges and then ranking. And then I'm going to use the weight as my ranking approach. And then instead of the green, again, I'm going to click on the box next to the color scale, use this grayscale that I have previously created, then click on apply. So now recenter my network a little bit so I can see it and get a sense for where this is headed. What we can do now, though, is since we resized things, I'm going to go ahead and rerun the Force Atlas 2 layout to prevent the nodes from overlapping one another. So just like with the Force Atlas algorithm, there's also a prevent overlap attribute that's associated with this particular layout algorithm. So I'll go ahead and click that and then run it again just to tweak the layout a little bit, and then click stop when it's done so that I'm not seeing any overlapping nodes. The next thing that I'm going to do is, again, like with the word co-occurrence network, I'm going to run that modularity algorithm to identify those clusters of nodes that are more densely connected to each other than other nodes in the network. So instead of being terms that co-occur very frequently in the word co-occurrence network, this time it's going to be clusters of papers that all refer to the same previous documents. So I'm going to go over to this modularity algorithm. I'm going to click on run. I'm going to use all of the settings as exactly as they are, and I'm going to click on OK. So in this case, it found 9 communities. And again, you've got the algorithm and resolution citations if you wanted to use those so that you know what you're dealing with. Then I'm going to click on close. So just like with the word co-occurrence network, it then added those numbers into the data set. So if we go up here to my data laboratory now, we have a number that corresponds to the modularity class. So in this case, we now also, though, have the article titles, we have the accession numbers, and we have the year published. So again, if we click on the modularity class, we can sort of sort these publications by the modularity class to get a sense for all of the publications that are assigned to a particular topic. And if we expand the article title a little bit, we can start to get a sense for what the papers are. So we see quite a lot of mentions to diarrhea here, rotavirus vaccines, the Malad cohort study I happen to know is about nutrition. So there seems to be a cluster around rotaviruses, childhood nutrition, diarrhea, those types of diseases. So that would correspond to a topic area in this larger publication set. Again, we can scroll down a little bit to look at some of the other clusters. You can see down here, cluster 3, I see there's geographical movements, combination therapies. So some of this might be about pathogen dynamics, jumps. So this may be modeling dynamics. So this may be modeling of virus disease and spread, virus emergence, maybe something in that area. Again, I would want to look through more of these publications just to get a better sense for what they have in common, and then name it based on what they have in common. So again, we need these article titles to be able to name some of these clusters, and so that's why we used that aggregate function file to be able to bring those article titles into the network so that we have them directly, and we can export this list of nodes to an Excel file or a CSV file that we can work with a little bit later on. So returning then back to our network, we can then color code based on those modularity classes that we just found. So I'm going to go back to the appearance box. I'm going to click on nodes and then color. And then again, I'm going to use partition rather than ranking here. And then the attribute that we're going to use is again going to be this modularity class that we just created. So I can click on apply here to apply the default color scheme to start getting a sense for where these publication sets are, the number of publications in each one of these topic areas, and a little bit about some of the relatedness between these different topic areas. So again, we could do some additional tweaking to reformat the layout. But from this point, it is, again, almost identical to that co-authorship network visualization where we go over to the preview settings, we export the visualization as an SVG file, clean it up in Inkscape, and then work with the final visualization from there. A couple of notes, though, that are a little bit different is that you do need some additional information to create a legend here. So you need to know not only what these colors are, but also what number those colors correspond to in your modularity class data set. So you need to know that this light pink color corresponds to topic 5 and not to topic, say, 0 or 8 or something like that. So again, in addition to exporting this table, you would also want to make a note of what colors correspond to each number in this modularity class attribute. There are a couple of ways of doing that. You can either say that 0 is pink, 7 is green, 5 is blue, whatever you happen to do with from there. You could also export this as an SVG file, open that SVG file up, and then put numbers just directly in here. Say that like this would be, you just put a 5 in here so you know that this is 5, this is 2, this is 3, this is 7, so on and so forth. But you want to do all of this before you close Gephi, or because if you close Gephi, you're kind of doomed. So you definitely want to make sure that you know what colors correspond to what numbers before you close out of anything here. Again, you could either do that as just notes to yourself. You could tweak an image that you export, however you happen to do it. But you do want to get that in addition to the node table in order to create that final legend to say that this blue color corresponds to this topic, which therefore is this topic's description that I actually give it. So you want to have quite a lot of information from this particular Gephi project to have those things available to you when you're actually creating the final visualization, the final legend, and so forth. So just as an FYI, you want to do a lot of this and make sure you have all of that before you close Gephi. It can be a good idea, though, to just go ahead and save this entire project as it is so that you can come back to it later on. So if you do forget something, at least you can reopen this entire project and it will keep all of these color schemes available to you, so you can go back in, and if you do forget to say what a color is or something like that, you can reopen the Gephi project and it will keep all of this information as is, so you can go back to it later on. So again, it's important to remember to have all of that information, or always save these particular projects so that you can come back to them. So as I mentioned, the rest of this process is very similar to what we've done previously, so I'm not going to recover it today. So that's essentially what I wanted to cover in this particular lesson. Again, there's quite a lot of additional things that we can do with topic analyses, or a number of different approaches to running topic analyses and so forth, but again, this is just one of the ways that we found very helpful in doing this kind of research for publications here at NIH and for other institutions.